Hi, it's Maria,
And Emily.
and today we discuss with Professor Silke Robatzek from the Sainsbury Lab about plant science and life outside the lab.
Yeah, it was a really fun conversation, I think, even though it was a bit shorter than our normal conversations.
Yeah, so hope you all enjoy our discussion.
So could you tell us, maybe introduce yourself and then tell us a little bit about your work and we'll go from there.
Yeah, I'm Silke Robatzek. I'm a research group leader at the Sainsbury Laboratory, and I'm interested in understanding how plants exchange molecular signals with microbial pathogens, in particular knowing that most of the agronomical important pathogens in plant that cause plant diseases are extracellular pathogens, so they colonize the extracellular space in the plant tissue. Now all the signal exchange has occurred across the plasma membrane, which gives a physical constraint. So it's this location and the changes that occur across the plasma membrane that is of interest in my research.
Were you always interested in plants in particular as a model system, or did you come at it from the pathogenic side?
No, I was always interested in plants, even as a kid. I was kind of like collecting leaves and putting them next to each other and asking myself, oh, they're all different. Are they only different outside or inside? So I always was curious to understand basically what makes up plant biology and what's happening inside. So hence I'm a cell biologist.
Yeah, we talk a lot actually about being cell biologists who work in plant systems. And I know from my perspective, when my friends or my family ask me, oh, so, how do I get my plants to grow better? And I think, well, I don't really know anything about plants, but I can tell you a lot about how the cells work.
Yeah.
But you said that you were interested in plants right from the get-go.
Yeah.
Yeah. So do you consider yourself more of a cell biologist or more of a plant cell biologist?
I would actually say by training, I'm more interested in plant physiology because it's the physiology that matters in the end, and cell biology is my particular way to look at it, just because also the way I'm thinking in pictures, so that kind of match how my brain works. But in the end, cell biology is a technique.
Right.
It's like genetics, like biochemistry is a way how you as a biologist would basically address your biological question. You have the same biological question and a biochemist would do this approach, cell biologist does this approach, molecular biologist does this approach. Yeah. But I think by training and the way I'm kind of like care about plants is more about physiology.
So do you only work with Arabidopsis, that is the model organism that all of us we are using, or do you do any other species or even crops?
Yeah, so I mostly work with Arabidopsis for the main reason, as a cell biologist, you always depend on genetically encoded reporters and stuff like this, and you depend on the mutants. But of course, we also use Benthamiana as a transient system. We do a little bit in tomato, and of course, I'm interested in the future of maybe also doing something in a grass model.
Yeah.
But mostly it's Arabidopsis just for, the materials.
The tools.
The tools.
Yeah. Do you find, this is one of the things too that we've been talking a lot about, is the importance to engage with society or government or the public in general to show that what we're doing is something that's going to benefit the community or people, and that seems to push a lot of plant biologists, but probably biologists in general, to really think about things outside of models like Arabidopsis.
More applied biology.
Yeah, more applied biology as opposed to basic fundamental research. How do you navigate that as a PI or as a scientist with particular interests?
I think the question that matters here is, how do you define what is a benefit to the society? Is the benefit something you're looking on a time scale in 3 years? Is it on a time scale of 5 years? Or is it on a time scale of 10 or 20 years? What we need in research is basically both working hand in hand together. So you need fundamental blue sky discovery-based research, because if you don't do this, you do not make major discoveries, right?
Of course.
And I think a really, really good example, what I like from plant biology, is Ola Bono's work on the tailend effectors.
Mm-hmm.
She has done that work, I think it was 12 or more years, classical genetic mapping and a pepper system to identify how all these tailends work, right? But now it's a super technology tool for engineering genes, right?
Yeah.
But it comes from a basic interest and understanding how Solanaceae interact with Xanthomonas bacteria and how Xanthomonas bacteria can cause disease. And I think we need this for fundamental big breakthrough discoveries.
Yeah.
And it's also, down to what's your talent and what's your potential as a biologist. I'm curiosity driven.
Mm-hmm.
I would compare myself with Alexander von Humboldt, I want to make discoveries. I'm exploring, I'm wandering around, right? I'm a discoverer. But then there's another set of personalities, biologists, that are much more interested in how can you use this knowledge to do that, which is more translation. I think we just need both together.
Mm-hmm.
Yeah, I agree. I think as someone who's also interested in kind of real fundamental understanding of how things work, and believing that really does build your foundation of knowledge, and without that you can't really apply anything because you don't know what those applications are affecting. But that also takes time, it takes failure, as much as it takes success. And how do you explain to those who aren't necessarily scientifically driven that the failures are also important? Or it's not even really a failure, it's just, well, we now know all of the things that don't work. Isn't that Edison says that he knows all the things that don't make a light bulb or something,
I think it's really difficult to make predictions in which research area there will be next breakthrough discovery. I mean, now everybody, particularly in plant sciences, is really happy about having CRISPR technology, because now you can research your questions in non-model systems.
Right.
You can use genetics, you can engineer traits, right? Everybody's really happy about this. How was CRISPR discovered? Right? It was a small subset of people that were studying antiviral defenses in bacteria.
Right.
Yeah.
And this is the fundamental research work, right? And now everybody, is so happy that we have this technology, because this basically is a door opener that makes a lot of, plant science independent of the model systems, and we can engineer traits, and that's great.
So when thinking about writing grants or talking to university departments who are interested in impact factors and all of the other types of kind of quantitative aspects of measuring your success, how do you approach those types of conversations? Do you just say, well, I measure my own impact and my own interests in these ways, and I hope that you do too? Or I know sometimes with grants are directed in a particular way, and then we kind of scramble at figuring out how our research fits in to that panel. Do you think that there's better ways that we can, moving forward as young scientists that might try, do we change it? Do we work better in the system as it is?
I think the current system has, in a way, shown that it works, and you can use the numbers game of citation index or highly cited researcher or age index or whatsoever, and there's some good things in that, but it also has some failures. So I think there's big room for improvement. And I remember. Years ago, I've been at a meeting of the Max Planck Society where a mathematician or a physician, I don't really recall this correct, I think he was a mathematician, has kind of told us about his enormous tour de force in a way, trying to publish his paper from his PhD in I don't know how many journals, and was always rejected. And then in the end, he was, over probably a time frame of 4 or 5 years, he was tired of that and basically just put it open source online, and then he used number of downloads or reads as his kind of metrics rather than journal impact factor. And lo and behold, you know, he got a EMBO Young Investigator grant. They say, well, this is super cool and we really like your work, right? So I think, you know, using the systems way to measure your success and your impact is good. But I think you also have to be creative of how you basically can demonstrate your own success. And probably the way you should ask yourself is, you know, have you added to textbook knowledge?
Mm-hmm.
Because in the end, you know, what, you know, is taught in school or in first year university students, that's textbook knowledge. So is your research making a change, making a difference to this? Has it added to textbook knowledge? And I think it's less about numbers of glossy journal papers.
That's a good advice.
Yeah.
Have like textbook knowledge. Yeah.
Kind of comes
It's like
back
a good
to
target.
The,
Yeah.
it comes back to the idea of kind of how fundamental research can be applied, not just maybe to making food more accessible, but to improving people's academic ventures as well.
Yeah.
Kind of backtracking a little bit, can you tell us a little on your influences as an early science researcher yourself, and kind of what helped you get to where you are today?
What helped me? I think, you know, maybe it's different now, but when I was kind of like, you know, becoming a plant scientist, I wasn't interested in a career at all. I was only interested in the science and in the research, and I was hungry for it and so curious. And my main motivation why to do a PhD was I figured, well, there's no other way I can do discovery-based research, so, you know, do the 3 years of discovery-based research and maybe you get a PhD degree or not, but I didn't care about that. Once I got my PhD, and same kind of like, I automatically kind of like got my next position. I figured, oh, there was this cool paper on flagellum. Maybe I go there, right? This really interests me, so that got me there. And then in my postdoc work, my initial project idea I wanted to do turned out to be absolutely not working out. So I started like a rescue project, being inspired from a multidisciplinary institute, listening so many times about EGF receptor endocytosis. I was like, maybe the flagellum receptor is doing it too, and that kind of like got me into this and then the next position. And so I just kept on going with not having as a goal a career, but always being just interested and curious on my science.
I feel like that's...
And I think I now, in my
familiar.
current job, I just got more susceptible to the career aspects and maybe got a bit blurred on focusing on my research.
Yeah, we interviewed Wendy Bickmore a few weeks ago, who is in Edinburgh, and she kind of had a very similar thing where she said, well, I didn't really know that I wanted the career that I have now. I just kind of fell into it. Yeah, you kind of just do it. And I think more recently for me, I don't know if Maria can say the same, but I feel like we're constantly thinking, how do we get to where we need to go? Am I on the right path? Am I doing all of the things that I need? When will I get to my career? When will I get from training or from being a student to being a real life grown-up, or, you know, whatever? And it seems that that is the process. It's just kind of being in it.
Yeah, but I think actually I disagree with this, right? Because you will get your job because you've done excellent research resulting in an interesting paper, right? And you don't get your job because you've done, I I don't know, a soft skill course, or you've networked well, or whatever. It is always down to the research, right? And the research will get your next job.
So, but I agree. I mean, I think that of course your work has to kind of speak for itself as well as for you. But there is a lot of kind of pressure to put on networking. You know, you have to talk to people to communicate your own work, and communicate the importance of your work, but also to find future employers or potential jobs there. So I personally would like to say, like, yes, I'll just I'll just do good work and things will come to me. But it seems as we get to be a more global scientific community, there is a lot of importance of also networking.
I agree with that, and I think it probably became more important in the recent years, because in the end, you know, I think there are many excellent, outstanding scientists, and a lot of job hires are given then on, you know, can I see this person as a future colleague with nice interactions?
Yeah, kind of personability as well as excellent research. Yeah.
And that you kind of like, if people get to know you as a person, you know, go to a meeting, present your work on a poster or in a talk, you know, but also join the evening activities, you know, go drink a beer, so they get to know you and see, well, you're also a nice colleague in addition to being an excellent scientist.
That probably brings the ideas as well of collaborations. It's not only, you know, of you having another job, but also creating a project. You know, as you said that you started and then you work on multidisciplinary areas, so probably you had some ideas from people that probably you wouldn't talk at the first place, you know, so it's not, maybe the word pressure has a bit of negative way on it, rather than it's like drives also, you know, collaborations and ideas and bigger things. Yeah, can you tell us a bit about your group then, like how many people you have, or like you have lots of students, and how you deal with all of basically how you manage the group?
I think my current group size is around 12, and I think majority is postdocs, and of course I'm interested in having people in my group that, you know, can work independently and can really drive their projects themselves. So of course they should work in a big team in my group, but I'm encouraging them to follow their own ideas, because I know a lot, but they might know something, you know, more experts in this particular area, or you know, they have an idea of which they like to pursue, and maybe this is the right idea to do, right? So I would encourage them. You know, when I was a PhD student and I identified this particular gene that I thought was so interesting, right? And then my supervisor was like so boring. And I was so disappointed. And then still I just kind of kept a little bit of work on this, and a couple of months later he came and said, wow, I think this is really interesting. So I think, you know, in the end, a lot is about, you know, finding the right balance, because between knowing and gut feeling. So I think also I appreciate that the people in my team are the experts on their topics, and they know more than me on their specific topic. So I encourage them to pursue their own ideas, even if I would be disagreeing with that.
Yeah, good.
Then of course I have PhD students on different stages, and, you know, of course they have, depending on which year they are more independent as well. I like to generate an atmosphere where everybody has their defined individual project, but it's easy to collaborate between them, so they can maximize and synergize, and they can have co-authorships also on other projects. For external collaborations, I also try to, you know, match, of course, with a specific expertise from the postdoc or the student, so they can also get more publications by co-authorships. I have a research assistant who runs the day-to-day business in the lab. We have weekly group meetings. Every 2 weeks I have one-to-one meetings with the people, and I run an open door office policy, which is when my door is open, you can pop in any times, discuss any matters, whether this is, some experiment has failed, or you have problems, or you need troubleshooting, or you had a super cool result. I also want to see that, because it also
make your day.
Gives me a gain, right?
Yeah.
To see like a nice plot that worked, or any kind of other issues.
How did you find the right number of people or the right size for your group? When you started out with your group, did you spend a lot of time on the bench yourself with maybe a technician? How did you grow your group to where it is, and how do you maintain kind of the right balance of people number-wise?
Yeah, I started out basically with a technician and a postdoc, and I think when you start your lab, the key is to hire your first postdoc. It must be the right hire. It should be a postdoc who is excellently skilled, but understands that this time you basically need to support the new growing group leader. So it's a lot of, building up, establishing, tools, systems, technologies, building a lab basically. So that first postdoc, is critical, to understand that this is about building up a group rather than, I want to have a Nature paper or something like that. And I was very lucky that I found such a postdoc. Basically also, when I moved from Basel for my postdoc to start my own lab, a PhD student was interested to move with me, and she stayed with me for, I think, close to 2 years, with the main aim building up the group. And then the more people you get, you specialize in the research project and so on. And it's true that in my first 4 years I was also still doing some work at the bench. I did a lot of radioactive works and a lot of microscopy works. But mostly, the technician kind of prepared stuff, and then together with her I did experiments. But now the bigger the group is, and you're not at the bench.
Yeah. Do you miss that?
Yes, very much.
Do you have any other things to, like, let's move a bit outside the lab and research and science. So do you have any hobbies or something that, you re-energize, since you are not doing all this kind of research on the bench that probably you like, but do you somehow feel this kind of emotions or feeling, this kind of positive attitude? Do you do something?
So for a long time, because I did this when I was a child and a teenager, I was quite attached to gymnastics. And I did quite a bit like active time, but then I kind of also worked as a coach, and then I became a judge, and then became international judge.
Wow.
And I also helped in selecting the GB team for the London Games and stuff. So that kind of like was really completely different world. And I remember that actually at the National Games some years ago in Liverpool, I was sitting there, on the podium training at the same time writing a review article. It's interesting.
True
because
work-life balance.
It's interesting because if your brain is completely busy with something, it frees up, barriers for your research. And I think this is why I figured you get many ideas very active in your brain. But now that I have a daughter, basically I had to kind of find compromises. So I can't do this anymore, and it's now, like, since 2 years, my daughter that takes up my free time.
Maybe she'll get into gymnastics, and then you'll be able to vicariously.
I have a kind of split feeling about it, because, I don't want to kind of transmit any ambitions on her.
let her be her own person.
Yeah, exactly.
Yeah. Nice.
Good.
Anything else?
Do you, I think we are almost
Yeah.
there for time, but it's something that we ask with Emily, if you use social media to network or communicate your research, and if so, how people can find you.
Yeah, I think I'm very bad with social media.
Yeah. Okay.
But you have
Right.
a website.
So the,
Yeah.
I have a website. I put on some YouTube videos, interviews.
nice.
I don't do Twitter.
Oh, with the new phytologist, right?
Yeah, the new
Yeah.
phytologist interview. I also actually did a movie myself. So I was basically writing the script and being the director of the TSL anniversary movie
Oh, nice.
that you can watch on YouTube, just to show like how is science at TSL.
Cool.
excellent. So I
Yeah.
guess all this information will be on the website. So if
Yeah,
people
yeah.
want to find it, okay.
Yeah.
Yeah.
Wonderful.
Nice.
But else I'm not good on social media, I think.
But still, you're doing a movie,
More,
so
yeah.
that's quite creative. So yeah, good.
Excellent.
Right,
Yeah. Thank you for talking with us a little
Yeah,
bit
no,
today.
you're welcome.
Great.
it's good.
So we hope that you enjoyed that conversation that we had, and you can find Silke's information on the Sainsbury Lab general page as well as her specific webpage for her lab. And you can follow Maria and me at Twitter or on Twitter. I'm ER_Larson_PhD.
And I am at m_Papanatsiou. You can also follow us on our institute's Facebook and Twitter. And see you next time.